Okay, this is chapter 4, part 2. We're going to start looking at a prokaryotic cell and look at all of the various structures that you may find in or on a prokaryotic cell. So we're going to start with external structures. We'll first start looking at this capsule layer. Now this is something that is optional. Some bacteria have capsules and some do not. So the more broad category for a capsule is called a glycocalyx. It just means sugar coat. It's basically an extra coating around bacteria that's made of polysaccharide, which is sugar, and or polypeptides. There's kind of two categories of glycocalyxes. If it's nice and organized and attached, it's called a capsule. If it's more loose and unorganized, it's called a slime layer. Kind of as that name implies, right? A layer of slime. The glycocalyx or capsule is really the term that I'm going to mostly be using throughout the semester, more than glycocalyx or slime layer. It is known to contribute to virulence. Virulence is the degree of pathogenicity, basically how infectious, how harmful is the microbe. So when bacteria have capsules, they are not able to be phagocytized by the white blood cells. And so that makes the bacteria more virulent or more pathogenic because our immune system cannot clear out the infection. What else? They're linked also to or involved with biofilm formation. As you can imagine, it's a slimy substance that's being secreted outside of the bacterial cell. When you get a whole lot of bacteria growing in an environment all with these slimy capsule layers, it contributes to that biofilm formation. So moving on, and let's jump back to that diagram real quick. We're going to talk about the structure here, flagella, these long hair-like structures that are coming off of that cell. So flagella, you've probably learned about them before. You know that they're for motility. In bacteria, they are structured a little bit differently than eukaryotic flagella. They have these three main parts, the filament, the hook, and the basal body, and they're made of a protein called flagellin. You can see how they're embedded within the bacterial membrane and wall here. This is for a gram-negative bacteria. And then this is how it's embedded within a gram-positive bacterial cell wall and membrane. Here's the animation.
The long whip-like portion of the flagellum is called the filament. Filaments are composed of many identical globular protein molecules called flagellin. Molecules of flagellin form helical chains around a hollow core. Certain pathogenic bacteria can be identified and classified by differences in their flagellar proteins. In contrast with eukaryotic flagella, no membrane covers the filament of a prokaryotic flagellum. The whip-like filament inserts into a curved structure called a hook. The hook is composed of a different protein and connects the filament to the basal body. The basal body is composed of different proteins than the filament and hook. The basal body is composed of a rod and a series of either 2 or 4 rings, which anchor the flagellum to the cell wall and allow the flagellum to rotate 360 degrees. You're viewing a flagellum from a gram-negative cell. A gram-positive cell would only have 2 rings in its basal body, both of which are attached to the plasma membrane.
Okay, and when bacteria have flagella, they can have different numbers of flagella and different arrangements of those flagella. This probably isn't the best picture to show peritracheus, but it's the one our book chose, I guess. But peri means around, all over. So peritracheous flagella is when there's flagella coming off of the bacterium all over, basically. They're covered in flagella. And again, it's not that obvious in that picture. Monotrichous means one, so just one flagella. It's polar. Obviously, it's coming off of one end. Lopho means a bunch, so Lophotrichus. And again, polar because it's coming off of that one end, but it's 2 or more flagella, right? Because 1 is mono, so anything more is LOFO. And then amphi means both. So here there's 2 on each end, but if you have flagella on each end, it would be amphi and polar.
Cells with a single flagellum are monotrichous. When the flagellum rotates counterclockwise, the bacterium runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Amphitrichous bacteria have a flagellum at either end of the cell. Bacteria with a tuft of flagella at one end are called lophotrichous cells. When the flagella rotate counterclockwise, the flagella bunch up and the cell runs. When the flagella rotate clockwise, they separate and the cell tumbles. Paratrichous bacteria have flagella covering the surface of the cell. Runs occur when all of the flagella rotate counterclockwise and become bundled, while tumbles occur when the flagella rotate clockwise and separate.
So, as you saw in the animation, flagella move by spinning, or I should say, flagella move the bacteria when the flagella spins, right? And that movement is called taxis, and they can be moving towards or away from something. If they're moving towards something, it's called positive taxis, and it could be chemotaxis if they're moving towards a chemical, or phototaxis if they're photosynthetic and want to move towards light. You can also have negative taxis, and generally that would be a negative chemotaxis where they may be moving away from a harmful chemical like an antibiotic or something. I think one of the animations mentioned about antigens. Now, antigen is a term we haven't seen yet, but we will definitely see in the future. It just means it's a foreign substance, something that is recognized by our immune system as foreign. So you can imagine this flagella, it's protruding from a bacterial cell. And if this bacteria is in our bodies, our immune system can see that flagellar protein as something foreign, as something that doesn't belong. So for example, in this strain of E. coli, O157H7, the H7 is referring to the flagellar protein that our immune system is seeing as foreign.
Motility is the ability of an organism to move by itself. The motility of a cell may enable it to move toward a desired environment or to flee from a harmful environment. Bacterial cells use structures called flagella, or in the case of spirochetes, flagella-like structures called axial filaments to enable this movement. Although flagella may appear to undulate back and forth like an eel swimming through water, they actually rotate 360 degrees, clockwise or counterclockwise. The basal body spins, much like the drive shaft of an electric motor, producing this motion. When the flagellum rotates counterclockwise, the bacteria runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Movement toward or away from a stimulus is called taxis. The stimulus can be light in the case of phototaxis or a chemical in the case of chemotaxis. In either case, the bacterium contains receptors which send signals to the flagella. In the case of positive taxis or motion toward the stimulus, tumbles become less frequent when the cell moves toward the stimulus and more frequent when the cell moves away from the stimulus.
So axial filaments are a special kind of flagella. They're also called endoflagella, and they're only found on the spirochete-shaped bacteria, those very long corkscrew-shaped bacteria. And the flagella is anchored at one end, as it is in regular bacterial flagella. The difference is the flagella is not protruding out in the environment. The flagella is actually wrapped around the bacterial cell, and it's contained underneath this outer sheath. So what happens when that flagella rotates 360 is that it causes the entire organism to rotate and to move in a corkscrew-like fashion.
Spirochetes are a group of bacteria that are spiral-shaped. They move in a corkscrew-like fashion, using flagella that do not protrude from the cell wall like typical flagella. Lyme disease is caused by a spirochete called Borrelia burgdorferi. Another spirochete, Treponema pallidum, causes syphilis.
Okay, so that was flagella. Moving on to other possible external appendages that bacteria can have. So remember that so far. We'll go back. So far we've looked at the capsule, which is an optional structure that bacteria can have. Flagella, which is an optional structure that bacteria can have. And then now we're going to look at fimbriae and then pili. And these are also optional structures. Not all bacteria possess these structures. So fimbriae, pretty simple. We just have this one slide on them. They're hair-like appendages that cover the bacteria. Often there can be hundreds to thousands of them. And the bacteria are using them for attachment to cells, basically. Like E. coli uses their fimbriae to attach to our intestinal cells. Pili, on the other hand, generally bacteria would only have one pili. And the major purpose of pili is for conjugation. The conjugation process is something we'll learn about in a later chapter, in chapter 8. But basically, what happens is this bacteria here is producing the pili. It extends it out, it contacts this bacteria, and it will draw that pili in and draw those bacteria closer and actually create a connection between the two bacterial cells. And now this bacteria will give some DNA to this bacterium through the pili. And that process of giving DNA is called conjugation. It's also called bacterial mating. Even though bacteria don't mate, they're purely asexual. But that's just the closest they get to mating, and therefore they kind of use that analogy. So because of that analogy, the pili often are also referred to as sex pili, since again, it's analogous to mating, I guess, in some regards. The other use that bacteria can find for their pili is this type of gliding or twitching motility, where they're basically just touching their pili onto a surface and springboarding off to glide, or if they continually kind of tap down on a surface, it can kind of just make them twitch and they get somewhere without having flagella, right? So I think they just kind of were inventive and thought of other ways they could use their pili when they're not transferring DNA. All right, now we're at the very important topic in this chapter, which is the cell wall. So looking back at this diagram, it's this brown layer here that if the bacteria does have a capsule, it's going to be below the capsule. But remember, some bacteria don't have capsules. So for some bacteria, this cell wall is their outer surface. The cell wall is made of a substance called peptidoglycan, and it definitely contributes to pathogenicity, as we'll be talking about as we go through the cell wall. So what is peptidoglycan? It is a polysaccharide, or I should say, I guess, a disaccharide. It's a polymer of a disaccharide. A polymer means a chain, and disaccharide is two sugars. So it's a repeating chain of these two sugars, N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM. And then it's cross-linked with polypeptides, which hopefully you remember what polypeptides are. Peptides are chains of amino acids. And polypeptides make proteins, right? Because that's what proteins are made of, chains of amino acids. So I know you were just dying to see the chemical structure of NAG and NAM, so here you go. I will tell you, you will not have to memorize chemical structures in this class. I can hear you cheering right now. But, chemistry is a prereq for microbiology, so you can see where there is definitely some chemistry in this class. So let's look at this more simplified diagram, though. So here you can really see that polymer of the disaccharide, of the NAG and NAM. The brown is NAG, the pink is NAM, and it's a polymer. It's a chain, right? And you've got multiple rows of these polymers of the disaccharide, and then they're cross-linked here with the polypeptide chains. And this is just a single layer, but they have multiple layers of this making up the bacterial cell wall. Especially gram-positive bacteria. Gram-positive bacteria have thick layer of peptidoglycan. That is their cell wall. Additionally, they have these teichoic acids embedded within it, two types, lipoteichoic and wall teichoic acids. And those play various roles for the bacteria, as you can see, dealing with charge, dealing with movement of molecules into the cell. So here's a diagram of the gram-positive cell wall. So if we just look at a simplified diagram of a gram-positive cell here, and if you were to cut a chunk out of it, out of the kind of external surface, you would see that the yellow here is the cell membrane, also called plasma membrane, cytoplasmic membrane. I tend to use cell membrane most of the time, but just know those are all used interchangeably. And the brown here is the peptidoglycan. Okay, and then you can see that here with that same diagram that we saw with the rows of the NAG/NAM and the cross links of the peptides. And you could also see those teichoic acids were in there too. And then we want to look at that in comparison to the structure of gram-negative cell walls. So gram-negative cell walls only have thin peptidoglycan, but they have this outer membrane that's made of polysaccharides, lipoproteins, and phospholipids. And this creates this periplasmic space, kind of this gap between the outer membrane and the plasma or cell membrane. And in that periplasmic space, that's actually where you find the thin layer of peptidoglycan. So gram-negative cell walls, because it has this lipidy outer membrane, it is more protective. It does give gram-negative bacteria more intrinsic resistance to our immune system, to phagocytes, to complement, which is part of our immune system, and to antibiotics. A major structure in the outer membrane is lipopolysaccharide, which contains, as the name implies, lipids and some different saccharides, sugars, right? Polysaccharide. O-polysaccharide, here again, is that bad strain of E. coli that was in the spinach. And you can see the O here, the O157, is referring to a specific polysaccharide that is in that strain of E. coli. Lipid A is an endotoxin. We're going to look at that more in just a minute. And then there is porins also in the outer membrane of the gram-negative cell wall because they form channels. The bacteria still needs to get stuff in and out of the cell. And therefore it needs these channels to do that because that outer membrane is going to be very exclusive and not let a lot of things pass through. So here is that cutout, same kind of image of a gram-negative cell wall. But notice the difference. The gram-positive just had a yellow on the inside, which was that inner plasma or cell membrane. And then it had a thicker brown, being that cell wall of peptidoglycan. But here you have a thinner brown, and then you have another yellow layer. That other yellow layer is that outer membrane. And you could see when you look at the more detailed structure that it basically is a phospholipid bilayer, which is what the plasma membrane is. So it is basically like an extra cell membrane, but now it's on the external surface. And it has embedded in it, it has this lipopolysaccharide, which has these polysaccharides and this lipid A that we said is an endotoxin that we'll talk about in a minute. You can see there's this periplasmic space here, and residing in that periplasmic space is this thin layer of peptidoglycan. There's the porin proteins as well. So since we already talked a little bit about gram stain in chapter 3, and now that we see a more detailed structure of the cell walls, you should be able to understand more how the differences come about during the gram staining. So remember that crystal violet is the primary stain, and it is a cationic dye. It is positively charged. It will stick to both gram-positive and gram-negative bacterial cells because they both have negative charges in the cell wall, in the cytoplasm. Now, don't get confused, like gram-positive, gram-negative, that it has anything to do with charge. It has nothing to do with charge, okay? They both have negative charges in their cell. Positive dyes are attracted. The positively charged crystal violet dye is going to stick to the bacteria and stain both of them purple. Then you add iodine, which is a mordant. Iodine crystallizes with crystal violet. It forms large complexes, large crystals that will get trapped in the layers of peptidoglycan. And once you add alcohol as the decolorizer, the gram-positive bacteria who have the thick peptidoglycan, that peptidoglycan gets dehydrated, kind of shrivels up, and that further traps in the crystal violet iodine. But gram-negative, because they have that outer membrane gets dissolved by the alcohol decolorizer. Now they're left with just that thin layer of peptidoglycan on the outside. It's not enough to trap in the crystal violet iodine. It washes out. The cells become colorless. What have we said about colorless bacteria under the microscope? Not easy to see, right? They're already so tiny, and then with no color on them, it's a pain. So what do? We counterstain them with safranin to turn them the pink-red color so that we could see the gram-negatives and distinguish the gram-negatives from the gram-positives under the microscope. So here's a side-by-side. These are some of the characteristics you would want to put on your Venn diagram for gram-positive versus gram-negative. Of course, you're going to want to, have some of the details of, here, starting here probably, with teichoic acids and things like that, as well as these characteristics here, okay? So the differences that were pointed out earlier with the flagella, the way they're embedded has to do with the structure of the cell wall. We have not talked about exotoxins, and we won't really talk about exotoxins right now so much, but we do want to mention those endotoxins again, because that was pointed out. I don't know which slide it was. Oh, right here. Lipid A is an endotoxin. Okay, and then you could see that lipid A here. So what does that mean? Well, toxin, as you probably know, they're not good, right? They're dangerous substances for us. And what it means here is that all gram-negative bacteria produce endotoxins because it's part of their cell wall. So that's what's important to know. As far as exotoxins go, we will definitely cover them more in the future. But what you can see is both gram-positive and gram-negative bacteria can produce exotoxins, and they're not part of the bacterial cell wall. So it really just depends on the bacteria. If they have the genes to make an exotoxin, they will make an exotoxin. But not all bacteria do. But all gram-negatives have an endotoxin because it's part of their cell wall. Penicillin and lysozyme. So penicillin, as you know, is an antibiotic. Lysozyme, you may or may not know, is a substance that is in our body fluids. It's in our tears, it's in our saliva, it's in our sweat. In both cases, and we'll see in more detail on a slide coming up, but these substances, penicillin and lysozyme, actually break down peptidoglycan. So you can imagine a gram-positive bacteria whose entire cell wall is peptidoglycan. When they're exposed to penicillin or lysozyme, their cell wall is going to get destroyed. What about a gram-negative? Well, they tend to be more resistant to that because they have an outer membrane that protects them, right? They have that intrinsic resistance. So if the penicillin or the lysozyme can't even get to the peptidoglycan in the first place, it obviously can't have an impact on it. So what we see is that gram-positive bacteria tend to be susceptible to penicillin and lysozyme, and gram-negative bacteria tend to be resistant to those substances. Now, I just realized when we're talking about that I missed a very important point on this very first slide. I'm pretty sure, I don't remember saying it, prevents osmotic lysis, protects the cell membrane, really should say protects the whole cell. Okay, this is a very important point. This is the function of the cell wall. So as long as the bacterial cell wall is not damaged, as long as it's fully intact, the bacteria will not undergo osmotic lysis. What is osmotic lysis? It means if the bacteria is in an unbalanced environment, and you may have learned about hypotonic, hypertonic, isotonic. We're going to visit those in a little bit here. But if the bacteria, especially is in a hypotonic environment where there's more water on the outside, what's going to happen? The water is going to enter the cell, and the cell, you would think, would probably burst. Happens to our cells if our cells are in a hypotonic environment, but our cells burst in hypotonic environments because we don't have a cell wall. We only have a cell membrane around our cells, which is not nearly as protective and as rigid as a cell wall. So as long as bacteria have an intact cell wall, they can take on water and they will not burst. But what happens if you expose this bacteria to penicillin or lysozyme or other substances that damage the cell wall? Now the bacteria will burst because their cell wall is damaged, and now they don't get that function of their cell wall. Let's see. We'll talk about what happens in a hypertonic environment in a little bit when we go look at those slides, because I think it'll help to look at the picture for that. But you definitely want to know that about cell walls in general. It doesn't matter if it's a gram-positive or a gram-negative cell wall. The function, the purpose of the bacterial cell wall for both types of bacteria is to protect from osmotic lysis. And as long as that cell wall is not damaged, they will not undergo osmotic lysis. But if the cell wall gets damaged, poof, they're going to burst. The outcome, as you might remember from chapter 3 of gram staining, is that gram-positive bacteria are purple, gram-negative stain a pink-red color, again, as explained here and in chapter 3. So that is important to know as well. So I'm going to stop this video here because we're approaching 30 minutes, and then we will pick up and hopefully be able to finish out this chapter with our third recording.